Hello, everyone. Good morning. Annyeonghaseyo. My name is Alan Griffith. I'm head of operations at Vector Builder. Today, I'm thrilled to discuss our innovative CliniVec service and its crucial role in accelerating cell and gene therapy. In this webinar, I want to set the scene regarding the global cell and gene therapy landscape, summarize some of the pain points we have in cell and gene therapy from the perspective of a contract developer and manufacturer. There's a lot of pipeline products entering the market now, and we need to find innovative ways to tackle these problems. And we found a unique solution in CliniVec. This is our agenda today. I'll briefly discuss Vector Builder and who we are. I will discuss the problem within the field. There's current issues and bottlenecks that we experience, a lot of our customers experience, and a lot of our competitors experience. I'll go to talk about CliniVec origins and how Vector Builder can assist and how we are uniquely positioned to help. I will then show how CMC fits in, which CMC is the manufacturing side of making a gene therapy product, and understand why that's important and why it's important for you, the audience, to know the differences. I'll then come to a close with some example case studies and data examples, which we've done before to bolster our CliniVec services to our customers. And then I'll conclude with some take-home messages and remarks and future outlook. Okay, so about Vector Builder, very briefly, who are Vector Builder? Vector Builder is all about gene delivery. We are on a mission. This is our mission statement: to be synonymous with gene delivery globally. Gene delivery, for those of you who may not know, is delivery of any genetic material using a vehicle, a machine to infect a cell that can impact a downstream genetic outcome. Again, we're focusing more so today on life science research. That is the area of biotech that we're involved in, but you can apply this technology across many different areas and sectors, as you can see listed here. Vector Builder, we look at ourselves as the first vector Amazon. We have over the last decade used this unique design studio platform, which has laid the foundation for all the work that we do with our gene delivery service. It was founded in 2014, so we've had our 10-year anniversary this year. Very successful business where we've now integrated a CRO, contract research organization, with the area that I'm involved in, which is the CDMO, which is the contract development manufacturing side of the business. So we've integrated both divisions into one complete integrated platform. And this assists researchers, academics, people in industry, and it allows people to gain access to these gene delivery tools at a very cheaper and faster rate. This is our global reach. Again, we are focused in 9 different countries. We have 10 different offices and branches opened. We are able to serve 90+ countries. We've delivered over 1 million different gene delivery solutions across all these jurisdictions and geographies. And we are affiliated with about 4,000 global institutes across academia, consortia. So very much a global presence, which we'll continue to expand. And then finally, just about Vector Builder, this is an important slide because we are wanting to show and emphasize we are all about vectors. A large portion of this presentation will focus on vectors. So these are 4 areas that we continuously break records in. We have 250,000 vectors produced a year. We look at various different sides of vector cloning, about 80,000 vectors a year. We look at viral packaging as well, which is where we not only provide the plasmid underlying sequence, but we actually manufacture the viruses as well. And then a large portion of our business is plasmid DNA preparation, which we do about 60,000 vectors per year. All in all, this pretty much means that we're coming up close to our 1 millionth vector milestone, a huge milestone within the sector that I don't think any of our competitors have reached to that number yet. So it's a very exciting point. But before I go on, I've just included a quick pause point here just to discuss what are vectors for those of the audience who may hear the word vectors a lot. I just don't want to assume that everyone knows what vectors are, just in case. Vectors, without vectors, we cannot build. We don't have a CliniVec service to take advantage of. Therefore, it's important to recap what vectors are. Vectors are simply just a vehicle to deliver foreign genetic material to cells. By virtue of that, they can be replicated, expressed, and interact with different host cell lines. This is very important. This is the fundamental of a vector. It's just used as a delivery tool, a vehicle, a van, a car. Numerous analogies can be made to a vector. It all starts with a plasmid. You have some sort of underlying sequence that is required for you to contain all your DNA elements, all your components for a therapeutic, for a gene delivery tool. You have to start somewhere. It starts with a plasmid sequence. On screen, I've showed pretty much a generic plasmid map, which shows the transfer vector and it shows a packaging vector. Every single client we have, of which we have 20,000+ clients, will have their own unique design, own unique needs and requirements for these genetic elements. This is very, very important to understand as well. Okay, so that's about Vector Builder. Onto the meat. This is the problem statement. This is the why. Why are we setting up CliniVec? There's numerous reasons. I'll discuss a few of them here. Due to the time, I can't go into deep, detailed descriptions of all of it. But before I explain CliniVec as a solution, it's very important to understand why there's a need for it. From a snapshot of the entire landscape, as I mentioned, yes, we have steady growth in cell and gene therapy. Everybody knows it's an emerging area within biotech. It's very novel, very new. It's always emerging into new and larger indications, but it still has a huge overall issue that everyone experienced. We see this large 90% clinical trial failure rate, and this is not for any particular reason other than you have a lack of efficacy, you have maybe unmanageable toxicity, poor drug-like properties, and maybe you've got lack of commercial needs. So they're the four main areas that cause a clinical trial to fail. What we've seen over time then is you see this valley of death region where you have this period between phase 2 and phase 3 where you see a huge regression in the amount of successful clinical trials. So for example, you can see across the board all the different years and quarters, you go from a 200 clinical trial number down to double digits of maybe in the 30 to 33 range. This is a huge failure rate. Again, this is something that we at Vector Builder really want to target. We want to get this poor attrition rate much higher. We want to invoke this better scenario for our clients. So I think this is important to explain then in a different way. We think of drug development in a very linear fashion, and we think this is what we imagine. We imagine we have an academic client, or you might be an academic user or an industry user. You perform R&D, you perform process development on your product. You manufacture a GMP batch, GMP meaning good manufacturing practice. This is a prerequisite for regulatory authorities. But in reality, what actually might happen is you would have a period of research and development, again, across the same timeline, but then realize that during process development, you might have certain hurdles or certain issues. And therefore, you need to stagger your process development workflow. This means you add layers of your process development. So as you can see, PD1, PD2, PD3, process development workflow 1, process development workflow 2, etc. This is often missed by a lot of our clients, where yes, it costs a little bit extra more money to do that at that stage. But if you fail to address the process challenges required for your designed vector, these poor design choices or poorly sourced plasmid materials can fail to meet the product quality required. So what we're trying to invoke is if you bring process development as part of CliniVec earlier, you're less inclined to fail later. And this is a point that I will raise time and time again, and I will try and explain it in a few different ways. I mentioned two bottlenecks. There is many more, but two bottlenecks that I think are very important to understand are the issues and failures can come from a design point of view, and it can come from a manufacturing point of view. So in the middle, you can see a typical swimming lane of a development path. You've got the usual R&D activities, preclinical animal testing, clinical testing, and then for very successful people, past the valley of death, you have a commercial development product. I'll start with the design. Often we encourage our clients to use our vector design studio, and this is where you go online and you can put all those elements to the plasmid that I mentioned. You can design your own. It's very important that people have the ability to design their own. But often then that means they can design it quite rapidly, not put much, I don't think, as much effort or thought process into it. And then what they normally go into is a screening procedure to do their proof of concept work. So POC, meaning proof of concept. Every therapy needs a proof of concept at the start of R&D in order to go forward into preclinical testing. But what we see then is if you compound issues such as a lack of candidate selection, where maybe instead of testing 3 or 4 products or plasmids or vectors, you actually test one. You bring this forward into a representative model. You might not have sufficient funding to do multiple candidates or multiple models. Then you're pretty much guaranteed, because you're only limiting yourself to one design, one candidate, you often can have safety and efficacy issues. And then at clinical, you have this emergency stopping point where you cannot progress. There is ways around this, but it's going to cost you a lot of money. It's going to cost you a lot of pain. It's going to cost you a lot of tears. And you want to try and avoid that early on. Now, I'll explain that a bit deeper in the following slides, but it's important to understand that that comes from a singular decision where you might only have designed one individual construct or vector for your therapy. Switching gears a little bit to talk about the manufacturing, the same idea. You start in R&D, you have a basic virus production method. You design your vector from us or from any other CRO. Then you start to manufacture it. You need to make enough virus in order to do your R&D proof of concept and to do your preclinical work. What we find after a decade of experience is we manufacture the virus for people, but a lot of our clients also manufacture the virus themselves, either using in-house kits or they purchase equipment and they do the manufacturing at small scale themselves. Because they're not a CMC or a manufacturing expert, this leads then to a year or two where they are really misunderstanding how the manufacturing, so the CMC, which is chemistry, manufacturing, and controls, is a fancy word for GMP manufacturing. Basically, that means then that you have built up this huge body of work, but you have no control over your process. In the field of cell and gene therapy, the process equals the product. So therefore, if you have built up a year or two of really not understanding your manufacturing, it often leads to a CMC failure during the clinical phase. And a CMC failure basically means a manufacturing failure. It's pretty much doesn't matter how you get to this point. You have either a design issue, safety issue, or a manufacturing issue. They all lead to the same place, the valley of death, where you either have to go back to the start, 2 years, 1 year, 3 years previous, or you try and remanufacture, you try and spend a lot of money doing extra analytics, and you can't actually get any further. So this is the two bottlenecks of cell and gene therapy. This brings me on to my next point. This is where all of these problems are everybody's problems. I decided after speaking to some colleagues, that a good way to explain this would be to design the perfect ATMP developer. So ATMPs is Advanced Therapy Medicinal Products, which is an umbrella term for cell and gene therapies. In order to be the best developer, you really need to look at being a medical doctor. You need to be a vector expert. You need to be a quality control expert. You need to be a CMC manufacturing expert. You need to be a good animal handling expert, an assay specialist, a document controller, a regulatory expert, and a device specialist. There is probably other attributes needed. As you can imagine, in the audience, or myself, or anybody in the field, me having been a previous drug developer myself, I simply do not have all of these attributes. These attributes are multifaceted, and they paint a larger picture. It needs a larger team. As you can see, multiple attributes are needed to be successful in avoiding those bottlenecks I mentioned. But this means you have to rely on large teams, which many startups, many academics, many early stage researchers simply don't have access to. What Vector Builder are doing with CliniVec is this program where we provide comprehensive solutions to address all of these challenges and needs, and make sure that all of you in the audience and all of the potential customers we have in the future have all of these attributes needed. CliniVec will hoover up all of the needs here and create a more suitable package for your therapy. And this is very, very important. Okay, so I think then this goes on to my next slide. Why we're uniquely positioned to help. CliniVec, it's, as you can imagine, clinical vector, short for clinical vector. This is why I think CliniVec is going to be successful. It's a service, as I mentioned, to help drug developers design these products quicker, better, and ultimately cheaper. I put the word ultimately underlined here because, as I mentioned in the swimming lane diagram, you have to spend a little bit more money on candidate selection upfront. That is so important because that will avoid you spending millions of pounds later on. A lot of people struggle to understand this. So what we're trying to do today is not 100% guarantee success if you do that, but get you to think about it in a different way. It's very imperative that researchers optimize their vector designs early, during the preclinical process, or before that, in order to conduct time-saving and cost-effective studies later on. Basically, in simple terms, it's a tool, and this tool can achieve 3 major milestones for you. It can implement your technologies to meet a target product profile, so a TPP. Without a target product profile, you cannot go into clinical trials. Secondly, you can optimize construct design for clinical applications. So as you know, you can have numerous designs. You can have N of 100 if you want. Very difficult to optimize that if you don't have the supporting infrastructure to do so. It also then verifies the manufacturing. So as I said, it optimizes the design and it optimizes the manufacturing. The 2 bottlenecks that I've mentioned previously, that is known now in the field. So basically, yeah, it's a consultancy service, and it's not a direct path to clinic. Everybody will have different needs. We can use a mixture of literature or in-house data that we've generated already to make decisions and recommendations that are going to add to your overall pipeline. At its fundamental level, it's a consultancy service that can bridge basic design with good clinical manufacturing, and it allows you to become battle ready. So this allows you then, instead of just being a person with very few of these attributes, you can actually become more of a superhuman, more of a person that's able to be battle ready going towards the clinical setting. Where does it come from? Where's the evolution of CliniVec? That's a question we get asked a lot. It comes from a convergence of factors, really. As an emerging leader in the field, I suppose we're uniquely positioned to be exposed to the headwinds and tailwinds within the investor community. Instead of being a typical CDMO, we actually keep very exposed in different circles where we know what's happening before it happens. We know what investors are thinking. We know what regulations are thinking. And also we have just released this new initiative called Dare to Lead, which basically it was born out of necessity, where we are trying to build a better future. We're trying to build better cell and gene therapy. And it's taking a more holistic view on thinking globally. Instead of being very transactional with our clients, we are trying to actually, be more involved, be more like a partner with you than a transactional person or a transactional organization who you pay for services. The only way the entire field grows and gets better is by being in a partnership with your trusted CDMO like Vector Builder. I've listed off some points here. I'll go through them quite quickly due to time. But the investor sentiment currently right now is that many venture capitalists do not want to invest in risky single design constructs. So this comes back to my point that if you just take one single design and you really only focus on that one aspect, you've got one shot at goal. You don't potentially give yourself mitigation. You don't have many shots at the goal. You're being quite restrictive in how you're designing your therapy. Investors don't like this. Investors really want to see that you've got more than one candidate going through your preclinical project workflow. But then also that's a bit of a pain point, because that means you need to spend more money on clinical constructs. But when you think about it, it will ultimately maximize the investor value in the long term. So these investors that are funding bodies, you might be an academic that needs funding from a government body, they will also ask you these type of questions. And it's important then that instead of maybe being confrontational about it, you can actually say, no, we've designed extra constructs, extra candidates, extra vectors intentionally in order to maximize value for your funding, maximize your VC money. So this is important. And then you can build up that track record for more indirect means. And that means getting extra candidates, okay? Moving on to the regulatory side, this is the second pain point. A lot of regulatory agencies have evolved because the field has evolved over the last decade. That means the regulatory agencies, the FDA, the MHRA, the EMA, have all evolved as well. 10 years ago, I've got products into the clinic. Doing what I did then, but doing it now would probably not be sufficient and not be acceptable going into clinical trials now. And that means extra sequencing, extra analytics, extra QC, spending more money on extra candidates. The whole landscape has changed dramatically. And what we're trying to explain here is that we at Vector Builder, because of our exposure and because of our positioning, we plan to align with all the evolving trends that will allow us to favor more predictive outcomes for our clients. The whole idea there, what is predictive outcome? It's basically combating recent safety concerns that are happening in the field now. They're evolving because we're seeing safety concerns happening because of poor manufacturing and poor vectorology design. So what we're trying to do here as part of CliniVec is make sure that we can combat both of these problems very early. And then ultimately boost candidate selection options for people. So if you do fail on one of those designs, you've got in your back pocket extra designs that you don't have to go all the way back to the start. You actually can start on the journey halfway through already. And ultimately, it's about de-risking your product at later phases, which not many people are concerned about. Phase 2, phase 3. We're trying to make sure that you are aware that you need to think about those decisions now in order to be successful. And then our Dare to Lead initiative is very important as well, because we want to think more globally and holistically. We want to do compliance. So this is going to be compliance by design. We design our vectors with compliance in mind. And that means instead of do-it-yourself cloning all the time, you would call our CliniVec consultancy service and we would talk you through it before you order your exact design. So basically it creates a competitive advantage for a lot of users. And ultimately, same again, it will de-risk your product at later stages. I mentioned vectorology a lot. It's good to pause here and just explain what vectorology is. Vectorology is basically all genetic components that encompass with inside a vector. So as you can see from the diagram, you've got a virus shell that's then packaging a certain configuration of DNA elements. These elements here, I've just used this schematic. It just shows and emphasizes that you've got 9 individual elements that typically make up a therapeutic product. The main message here is that if it can be modified or changed, then it can cause an effect, a therapeutic effect, and this effect could be good or bad. It can be deleterious. And therefore, each element must be well understood and characterized. It must have a purpose. If it doesn't have a purpose and it has come from a different source, or you might have sourced it from another academic institute or a collaborator, and you cannot explain why you have got one element in it that doesn't need to be there, then it should be removed. And this is also part of CliniVec. CliniVec, I don't want people to think it's just an immediate design solution, but also some of our users give us a plasmid to start with, a precursor plasmid. It's important then that as part of our CliniVec service, we also understand the existing plasmid design, not one that maybe we've designed for you, because that's one part, but we also can evaluate your existing plasmid. And this is so important that we will be able to tell you if you should modify it or if it's actually okay to proceed. This is the 2 different ways you would look at the CliniVec at the vector stage, the design stage, because you can use an existing precursor or you can design it from scratch. There's benefits to both, which way I will explain in following slides. So this is basically it. You have, in summary, you can do there's 2 approaches to our CliniVec service. You can have a basic CliniVec or an advanced CliniVec. If you utilize our basic CliniVec service, we can guarantee that you won't have any sequence scrutiny from the regulatory agencies. Doesn't mean that it's going to be efficacious. Doesn't mean it won't have manufacturing issues. No, we cannot guarantee that. But what we can guarantee is that the design doesn't have extra elements in there, doesn't have extra superfluous regions or elements that may cause issues or may cause safety events. And I've chosen 3 examples of this. A lot of vectors use ampicillin as an antibiotic resistance gene. This is actually now coming to a point where all the regulatory agencies are advising users to move to kanamycin or neomycin and to get away from beta-lactam hypersensitivity. Because ampicillin is associated with beta-lactam hypersensitivity, it is generally considered a safety concern. This is something that the field has learned over 10 years. Again, not to worry anybody who has an ampicillin backbone in their vector. 10 years ago, this was acceptable. But now, the last few years, the regulatory agencies have come down hard on this. And it's very important to understand that you should always, at least at the minimum, have a kanamycin backbone rather than an ampicillin backbone. The same can be said for WPRE, which is the Woodchuck response element, which boosts gene expression, it boosts RNA stability. There's now different versions of the WPRE. The wild type version has been shown to have oncogenic properties. You see oncogenic issues happening in non-human primate studies, in macaque studies, and in some rodent studies. So our solution to this very basic suggestion is to migrate the WPRE, if you need it, to a mutated version or a truncated version of the WPRE. And this is basically, by doing those 2 things, you're already making your vector more clinically compliant. And then I use the F1 origin of replication here as a very simple example where the F1 origin is often contained in a lot of precursor plasmids from people that we receive, and it serves no purpose in the downstream applications. Will it cause a safety issue? Probably not, but it comes back to my point. If it's contained within your vector and it doesn't have a purpose or a clinical purpose or a therapeutic purpose, then it should be eradicated. It should be removed. So they're the three areas I think that our basic CliniVec service. We do this for the majority of our clients right now. But then if you really want to go the next level, you have a bit more budget, you have a bit more time to do extra candidate selection, we can look at all of this, these three areas, these three pillars: extra characterization, extra safety profiling, or extra performance. It's not all about safety for CliniVec as well. It's about, we want to boost performance, because if you have better performance, you're going to have a better therapy. If you have a better performance, you might have a lower dose requirement, which would mean that the amount of virus that you need to manufacture is a lot lower. This means you save a lot of money when you're doing GMP manufacturing. I won't list out all of these benefits. It's here for you to look at. If you want to discuss any of the advanced workflows, please reach out to support. This is the CliniVec typical workflow. It will look slightly different to everybody, but this is the general concept. It comes from different scenarios. Basically, we want to give you, the developer, a checklist. We want to cross-examine different things. It starts with an introduction meeting. You talk to one of our territory managers or application scientists. We understand a few different things. We understand, do you have proof of concept already? Do you have funding? Do you need data for funding? Do you have an established animal model? All of this allows us to understand if we can qualify you as a CliniVec user. And again, this is something we work with you. And if you're not a CliniVec user yet, that's okay. We will take our time with you. We will talk you through possible scenarios in order for you to get on your journey to the clinic. Then normally we look at a technical presentation, and we would present your design, your multiple designs, if you want to have multiple designs, and we would sit down and discuss with the client maybe your entry point and the candidate expansion. So we've coined a term called price per clinical candidate, which is PPCC. This is a tool or a metric that we're hoping will catch on with some of our clients, that in order to request more candidates, you would have to maybe expand your price per clinical candidate. And then this will allow you to ask for, or include it as part of your grant applications, talk to your investors about it, talk to your colleagues about it, and see if you can actually generate more candidates. Ultimately, it all goes down into, can we generate a suitable design for you in the middle here? Can we design it? It can be done with an in silico approach, very computer-based, literature-based, or we can do that, plus we can add functional testing approaches to it as well, where we would do comparative analysis, sequence efficiency testing, manufacturability assessment, deeper characterization in how those constructs behave in bioreactors, flasks, how they behave regarding quality, like the quality of the capsids that they make. Do they generate a lot of empty versus full capsids? All of these things can be done at a functional level in real time. Ultimately, to get the goal where you want to have suitable performance. What is suitable performance? Does it link with your target product profile and the way you've built your design of your product? Then you would agree a lead candidate, and we would both agree that you don't need to do any more design, you don't need to do any more vectorology. And then that's your starting point where CMC, our manufacturing process development, starts. So if you can imagine the schematics I showed previous, where you had the process development workflows, this is at the right-hand side of this workflow, where you have process development and tech transfer after you've agreed your lead candidate. Of course, then you go into scale up, large scale manufacturing, which is a topic for a different day. Taking the product to clinic is not easy. I often mention that these vectors come from different sources. They can be academically sourced, they can be kind gifts, often have poor traceability. This is important for us, that we want to try and understand this. Over the last decade, we've understood that the main gating factor for safety and integration is actually vector design. We're also very aware that our users are worried that how do you know how many vectors you need to create? The answer is we don't. You can have numerous iterations. The iterations could go on forever in reality, but the goal of CliniVec is to try and make sure that you minimize the amount of iterations you need to do, because everybody knows it's the best approach to bringing a product to clinic now. And this is so important as well. And instead of looking at it as extra work, I think we try and set the scene, the mentality, the mindset change should be, this is actually a competitive advantage, because if you're going to the clinic and you see the regulatory authorities are asking you questions, or your investors or your grant agencies are asking you questions, you can say, we've de-risked the path to clinic by having more vectors designed and better sources of our vectors. We have gone to Vector Builder and done de novo synthesis. Therefore, we have no traceability issues as part of our vector. This is very important as well. And again, yeah, just moving on then, why I've mentioned CMC and where does it fit in? When you have a product during this design stage, it's important that you understand, can it be manufactured? The main message here is, if you don't allow for manufacturing scale and you don't know about the performance of your product at later stages of manufacturing, well then, unfortunately, you may not have a product. You might have a product that works really, really well. It's very efficacious in an animal model. You can manufacture it in a small scale, either at a CRO or in-house in your own lab. But because you've never subjected it to large-scale manufacturing, you don't know how it will perform at a scale-up stage. You have, as you can imagine, you have impurities such as host cell DNA, residual proteins, empty capsids, partial capsids. These can all be amplified or magnified to a point where the specifications for a given manufacturing batch may not be met. So therefore, it doesn't matter how good your product is. If you can't manufacture it, you don't have a product. And this is a problem that a lot of people in the field suffer. And that's why I mention it as a bottleneck. It's a huge bottleneck. Manufacturing, we're not worried about manufacturing. The amount of clients and CliniVec clients that we speak to, we're not worried about manufacturing. And then we said, that's okay, it's your decision. And we let them go. And then 1 year later, we have a problem, Alan. This is not manufacturable. This is why we're trying to make you aware of it now, that we can accommodate and do some feasibility work very early, so then you don't waste millions of pounds later. This is very, very much an issue, and it goes unaddressed time and time again. It will manifest all the time as well. So yeah, this is what I mean by manufacturability at scale. I've just used the bioreactors here. Of course, you would maybe in a commercial setting use the bioreactor all the way on the right, which would be a 2,000 liter SDR. Again, you're talking about £1 million per batch campaign here. Obviously plus and minus, but that's why these products are so expensive to make, is because they're very difficult to make. But as part of Vector Builder's CliniVec offering, if we actually explore how to manufacture it easier, we're bringing those cost of goods way down for you at a later stage. And we believe that there's no other CDMOs that are offering this service. Again, this is just saying more or less the same point around the evolution of CliniVec. We've got a huge funnel. Why can we say all of these things? We've almost made 1 million vectors. That means we can understand and leverage all the information, all the knowledge, all the expertise that we've built up over the years, and apply that to building these different cars, building these different vehicles. So I think over the last 10 years, there's been a lot of manufacturers making a car. Again, it's got 4 wheels. It's going to get you from A to B on your journey, and it will be acceptable. It will be okay. It will work pretty okay. What we're doing with Vector Builder is we're trying to leverage all the expertise of these 1 million vectors and make slightly better cars, slightly better engines, slightly better ways in order to get your product, from A to B in a much less stressful and problematic way. Yeah, I think this is good to show. This is how we design our vectors. This is our DIY design studio. It's very important to understand that you can go online yourself and design it. And then we've included a lot of extra information on our interface now, where you can reach out to the CliniVec team, you can reach out to myself personally as well. And instead of just normally outputting a file that you can render straight away, what we've done is put CliniVec as a bit of a stage gate, where we would make sure that instead of you ordering and potentially ordering based on you not knowing all the attributes you need, we would allow Vector Design Studio to actually catch you at the right time. So then that way you're ordering it properly, you're ordering it on time, and you've got consultancy before you order it. And this is very, very important. Again, the Vector Design Studio, this is how we've built the business over the last 10 years. It's all backed by integrated design, which marries vector design with pretty much a seamless vector interface. You can click, drop, drag and drop, the ability to order online after a few clicks. Again, it's the Vector Amazon style that we are going for. We have thousands of users again, and at the same time, we really emphasize here that we don't want to oversimplify this design. If you're worried about anything of what I've said today, please reach out to our CliniVec team. So why is it important then to link this data during your IND? So your IND is your investigational new drug. You need an IND to go into clinic. But as I mentioned, it's important, and this is why I'm repeating myself a few times. It's very easy to overlook it. During the swimming lane, again, the typical development path, very simplified. This is what we're trying to do. We're trying to get people, instead of having a later entry point, we're trying to bring the entry point for your proof of concept earlier. So then that way you invest more of your time. And when I say budget, I mean your time. Your time is money. So your time and your budget here. Ultimately, your cost of research goods is actually manageable at this stage. Anything before clinical is going to be an order of a magnitude lower in cost for you. So I think it's very important to understand that if you invest your money early here, you save money here. And this is not insignificant. I mentioned £1 million per batch. Again, I'm being very simplified, but generally then, if you increase the effort and money on your price per clinical candidate, again, using the terminology I used earlier, it's a lot easier then to manage your costs later. And this is called runaway costs, where you've got the investor locked in, you've got your grant, all of a sudden you didn't accommodate for many candidates, and then you have to actually spend way more money than you anticipated on your budget, because it might not have the best design and it might not have good manufacturing. Very important to understand these points. And then ultimately, if you think about it from two different sides, you've got a backbone side and a payload side. And I think a lot of people think of the vector elements individually, which can be useful. But if you take anything away today, if you think of it as two main parts, two macro parts, that you've got the left-hand side of your vector, which is your backbone, and your right-hand side, which is your payload, which is your packaged therapy. Both of these sides impact the final vector profile. The SISPQ, which is the safety, identity, strength, purity, and quality, which is a prerequisite for a CTD document. You have to explore the impact of all these sequences on the SISPQ. And I think a lot of people just focus on the right-hand side of their vector. They don't actually believe that the backbone side impacts it, but I'll go to explain why it does in a few slides time. Again, just want to highlight the difference between the costs. So when you do later stage GMP manufacturing, you need to go through process characterization, which, remember, your product equals your process. So basically you need full-blown product and process characterization. And this is taken from the regulatory agency, where I've taken it from exact wording, where they want you to identify and quantify all significant resources of variation. And this variation comes down to the starting materials. And if you think about it, what is your starting material? Your starting material is your plasmid. So any variation or intrinsic variation in your starting vectors or your starting plasmids will mean that it will impact your process characterization later. And if you have problems with process characterization, well, that's where you end up losing lots of money later. Okay, so then, yeah, now that's pretty much CliniVec at a consultancy level. Now let's show you some kind of case studies and example data, which is important to understand what I mean and put a context to what I've said, obviously. First case study, I mentioned earlier that plasmids or vectors can come from different sources. So yeah, they can be sometimes a gift. An academic institute gives it to another academic institute. That's okay. That sounds good. But there's been cases, and I've used one of our customers' examples where they had a kind gift from an institute. They spent a lot of effort on doing preclinical animal testing. They manufactured the virus with us in order to put them in non-human primates, and then realized that the virus was suboptimal. It had very poor packaging. It was very difficult to manufacture because it was oversized, all of these issues. So the story here is I want to catch people and understand that there is a journey from an academic mindset to an industry mindset. Again, not saying there's one way is wrong and one way is right. It's just making you aware that if you do have potential products destined for the clinic and you've got it as a kind gift from somebody, it's very important to understand where it's come from and has it been traced? Is it compliant already? Often we show that it's not compliant. And a good way to do this, I think, is to explain you can create it from scratch. And when we design it from scratch, we can clone it. Or a lot of people now are wanting to do de novo synthesis from the start. So it's the first step to compliance. If you design a vector, instead of cloning it from different sources, you just you make it, instead you do it from de novo synthesis from scratch. The regulatory agencies love this. They really, really love this. The fact that you've put every nucleotide is designed there and it's come from a chemical synthesis means that's so important, I think, to the building the safety and the confidence around the vector. And so this was a classic example of old versus new, where you've got a suboptimal vector, and okay, you might have the same efficacy, you might have the same performance, but now it's a lot safer, it's better manufacturing, it's got better QC ability, and that's important as well. Case study 2, moving quickly. This is a case for the backbone, where we designed and optimized the backbone and chassis of our plasmids, where the backbone is, as I said, half of the whole design of your therapy is the backbone. It's important to optimize this for manufacturing. As you can see, we looked at AAV, we looked at LV, lentivirus, we looked at plasmids, and we looked at HR donors. The whole story here is that we were increasing manufacturing by improving process yield, removing the impurities, and increasing the production scale. So as you can see then, by adjusting the backbone, we're able to optimize it for manufacturing. Therefore, we can use this backbone in many of our clients' projects. And very quickly, thirdly, this is a lentivirus project where we had a typical workflow, a very standard 3 to the 10 to the 7 transducing units as your yield. What we did then do is we subjected it to prime analysis, and we included CliniVec PD and CliniVec design modifications, and we were actually able to boost functional lentivirus titer up an exponent, up a log, up to 1.5 E10. And then increase the amount of positive cells that we were going. This was part of a CAR-T experiment. So we were able to boost the amount of positive ratio of modified cells by having better functional titers. So again, it shows that it's not just AAV, it's not just plasmids, but we can also look at lentiviruses as well as part of CliniVec. And yeah, so this is the last slide or two now coming. So hopefully everybody is still awake. This is the another pause point where instead of doing standard QC, we're also looking at CliniQC. It's a hidden part of drug development where, you know, I'm always saying we've got the left and right side. Why do small changes lead to such drastic differences in a product? Well, if I had to use any schematic, I would use this schematic here on the bottom left, where you've got your full capsid containing your therapeutic construct. But in reality, you normally have 4 different populations of your vector, any virus vector. Again, I'm using AAV again as an example, but you get this phenomenon known as reverse packaging, where the virus assembly and packaging can be so random that you end up getting packaged pieces or partial parts of the DNA. These are bad. These are known as product-related impurities. These are essentially, these need to be eradicated. And as part of our CliniVec service, we can look at ways to improve packaging efficiency. Packaging efficiency is its own parameter that we try and test early. We look at GC content, splice sites, removing cryptic splice sites. We look at removing termination signals. All of these have been shown that can boost packaging efficiency and reducing the backbone packaging, which is the reverse packaging, which is essential. Any virus piece or any virus particle that doesn't contain your full therapeutic gene is considered a product-related impurity, and that's so important to remember. So yeah, and additionally, I think this just goes on. This is pretty much saying the same thing. Why is it concerning? These empty capsids or partial empty capsids, they have no therapeutic benefit. And from a safety perspective, that means there's a huge risk of an immune response or getting some sort of SUSAR event or safety event that might happen because of that. So therefore, this illegitimate packaging can be concerning, especially if you're packaging maybe whole cell DNA, an oncogenic portion of the WPRE, for example, that I mentioned. All of this can be unintentionally delivered to the patient. This is not what we want, and this can all be avoided using our workflows. This is more or less the same thing, but I'll move on because of time. And more or less the same thing here. Again, concluding remarks and future outlook. This is just to conclude. The gene delivery market is actually going from strength to strength. We are uniquely positioned. We want to ensure that our clients are making the best possible decisions. If you're making the best possible decisions, it means that you'll be successful. And if you're successful, we'll be successful. And not only in terms of, I think, optimization and cloning, but we're also trying to deliver better genetic medicine. We're partnering with a group called LANTU, which are allowing us to focus on Menkes disease. I think our most recent press release showed that we've actually manufactured a product for Menkes disease. So we're really passionate not only about doing all the manufacturing and design work, but actually making a difference in the clinic as well. So this is very important. I think we're going quite well. We're quite well positioned here that we'll keep this pendulum moving in the right direction for the next 10 years. And then, yeah, finally, I think this is good to know that we are building a mRNA facility. So we're building a facility to stay with the emerging trend of mRNA. And I think we're just going to actually build this now, and it'll be coming up operational in 2025. So that means that not only will we have CliniVec, but we will also have mRNA. Stay tuned for this. And yeah, so then these are our take-home messages. CliniVec will mean something different to everybody. That's okay. It's now known that optimal design and better design is better for therapeutic efficacy. So therefore, optimizing design is more important now than ever, and is starting to become a prerequisite by regulatory bodies. There's consortia groups being set up focused on this, and we're getting a lot of consortia groups asking us about CliniVec in order to see, can we support them even more? So this is very exciting because it means that we're not the only ones trying to say this story. Other people will start to say this story, but we are the first to raise it and first to actually bring it to the surface. And then finally, yeah, we believe that any element has the ability to be experimentally tested for better functionality. Again, we've built 900+ thousand vectors. We want to apply a checklist and make sure that you have the best vector for your therapy. And yeah, there's just some snippets that here's the checklist that we want. We want to have you less deviations, more shots on goal, show your investors you've selected wisely, you have backup candidates if something goes wrong, and then yeah, you'll have the best efficacy for the best product. So yeah, that's in my summary slide, and I want to thank you all for your attention. Again, we've got my other esteemed colleagues Connie Rich and Brady Strittmatter. They also have done brick webinars. Please check them out. Thank you for your time today. Hopefully the information will convince you and potentially future researchers that you can utilize our CliniVec service and generate your wonderful projects with us. Thank you.